Good day, everyone. Welcome to the Learning Biology Through Questions, Chapter 4 Enzymes. I'm Mr. Ong from College Tingkatan 6 Seri Putra. In this video, we will discuss about the free energy diagram, induced fit model, enzyme kinetic, cofactor of enzymes, how temperatures affect enzymatic reaction, and the role of components in a biosensor and how a biosensor function. This is a question show that a diagram of free energy for an enzymatic reactions, and they're shown in graph below. What do A, B, C, and D represent? Okay. A, B, C, and D. So A is the energy processed by substrate. Okay, energy processed by substrate. B is the activation energy for reaction with enzyme or of enzymatic reactions. Whereas C is the free energy change. Okay, you must have the free energy change. You cannot just say energy change or overall energy released from the reactions. The symbol is delta G, not delta H. Okay. D here is the activation energy of reaction without enzyme. Okay. So next question, what is meant by activation energy? So this is definitions question. Activation energy is the initial energy required to start a reaction. Okay. Or the initial energy required to overcome an energy barrier in order to start a reaction. If you don't want to use initial, you can use the minimum energy required and so on. What is the effect of temperature on the rate of enzyme activities? The rate of enzyme activities increases as the temperatures increases until it reaches optimum temperature. So for this one, when we go through the temperature, the effect of temperature on the rate of enzyme activity, normally students, they will only write this, the first part only. They forget until it reaches optimum temperature. So this is compulsory, okay? That means when you write the rate of enzyme activity increases as the temperatures increases until it reaches optimum temperature. Okay, because beyond the optimum temperature, the rate of enzyme activity or the rate of enzyme activity will be decreased as the temperatures increase. Okay, next question. What happens in the transition state for the enzymatic reaction? So during the transition state of the enzymatic reactions here. Okay, so the answer is the enzyme substrate complex form during the transition state. That means in the transition state actually is where the substrate, the specific substrate bind to the enzyme at the active site. Okay, so I must say the active site very important, it must be emphasized here. Then to form the enzyme substrate complex, so at the transition state, then obtains enough energy. Okay, obtained enough energy. So that means the transition state, the enzyme substrate complex, they obtained enough energy or sufficient energy to break the bonds in the substrate so that new bonds in the product can form or can be formed. Okay, so this is how we answer the structure questions. So here the importance here is enough. That means they have enough energy because we talk about the energy level here. All right. If I modify the questions, say into based on the induced fit model, describe what happens in the transition state. I mean, what happened in the transition state? Okay, based on the induced fit model. So the answer will be like this. The substrate, Okay, say the specific substrate binds to the active site of enzyme. In STPM, you must mention active site of enzyme. Okay, to form an enzyme substrate complex. Then the binding causes the enzymes to change its shape slightly. This is what we call induced fit, so that the active site enfolds or distorts the substrate and hold it in place. Okay, so they change the shape slightly, and also the shape of the substrate also changes slightly and becomes unstable. This is what would happen, what's happening in the transition state. So this enhances the ability of enzyme to catalyze the chemical reaction between the substrate and enzyme. Then, so that the substrate can form the product. So this is what happened in the transition state, and then we just applied the induced fit model, where they say that the enzyme changed the shape slightly, and also the substrate also changed the shape slightly. Okay, and then at the transition state, the enzyme substrate complex is unstable. Okay, that means that the substrate becomes unstable, so that the bonds in the substrate can be broken down, and then the new bonds is formed in the product. This is you can relate to the previous question here. Okay, the previous question is just a simple only. Okay, they say how is the enzyme substrate complex that obtain the enough energy to overcome this activation energy, okay, the energy barrier, so that the bonds in the substrate is broken down, and then the new bonds in the product is formed. Okay, and then here we just add in the induced fit model. Okay, so that the student see whether the student can apply the induced fit model into this, what we call the binding of the substrate to active sites of enzyme. So next question here, this one is talking about the Michaelis Menten plot. So name the curve X, Y, and Z. So X is the rate of enzymatic reactions without inhibitor. Okay, without inhibitor. And then Y is the rate of enzymatic reactions with competitive inhibitor. And Z is the rate of enzymatic reactions with non-competitive inhibitor. So this one very clear lah. Explain why the shape of the curve X is as such. Okay, X at low substrate concentration. That means at low substrate concentration, the rate of reactions increases rapidly. Okay, very fast, rapidly, as the substrate concentration increases. Because the question said explain, so because more active sites of enzymes are available to bind with substrate. Okay, then the rate of reaction then slow down. Okay, it slow down. Okay, it slow down and reaches maximum rate at low substrate concentration. Here still considered as low substrate concentration, because this is the for the curve of the reactions without inhibitor. Okay, so when they reach maximum rate, so this is because all active sites, okay, of enzymes are saturated, okay, or occupied with substrate. Then the enzyme concentration becomes limiting factor. So that means once they reach the maximum rate of reactions, the enzyme concentration becomes limiting factor, not the substrate concentration. Before they reach the maximum rate of reactions, the substrate concentration is the limiting factor. Okay, for the curve Y, okay, curve Y here, so you must mention competitive inhibitor. Okay, cannot just mention inhibitor. So you must say competitive inhibitor has similar structure. Okay, important, similar structure to the specific substrate. So the competitive inhibitor competes. You must say compete here with the specific substrate for what? For binding to the active site of the enzyme. Not binding to the enzyme only, binding to the active site of enzyme. Thus, the rate of reaction is lower. Okay, is lower than curve X. So we compare, right? Then a higher substrate concentration, okay, a higher substrate concentration, okay, higher substrate concentration can overcome the effect of competitive inhibition, as more substrate can bind to the enzyme to form enzyme substrate complexes. So this part is to explain why the higher substrate concentration can overcome the effect of competitive inhibitions. Or you can write by adding more or higher substrate concentration, the competitive inhibitions can be overcome and so on. Okay, so for this explain one, you must write competitive, that means specific terms. Okay, then the next one is talk about the curve Z. So for Z here is non-competitive inhibitors binds to the enzyme at site, okay, at the site other than active site, okay, at the site other than active site. Then the non-competitive inhibitors does not compete, okay, does not compete with the substrate for binding to the enzyme, okay. Then the binding of the non-competitive inhibitors to the enzyme changes the active site. Yeah, they bind to the, because here we already mentioned as the at the site other than active site. So here they will change the binding of the non-competitive inhibitor to the enzyme. Let's say you want to here to the active site of the enzyme also can. So changes the active site configuration. And the active site configuration. Let's say if you don't want to use configuration, you can use change just the active site shape or the shape of the active site. Okay, the substrate has no longer binds to the enzyme. Okay, then the maximum rate of reaction is lower than those in curve X and Y. Here you also can add, say that the increase of the substrate concentrations would not increase the maximum rate of reactions. Okay. Which curve shows an inhibition that could not be recovered by the addition of more substrate? So here's curve Z, curve Z. Okay. So for the next question, year 2020, last year question. I modified the question a bit lah. Okay. So the action of the catalyst with and without the presence of inhibitor are shown in the graph below. Okay. I changed the unit here. Okay, then I add two questions here, three questions later. The first extra question is derive the Lineweaver-Burke equation from the Michaelis-Menten equations given. So the givens here, this one, okay, V is equal to V max times substrate concentration divided by the Km plus substrate concentration. So in order to get to derive the Lineweaver-Burke equation, so we want to 1 over the equation first. So 1 over V, so this one upside down. Then after that, you can get it. Okay, that this one, here we split it. Then after that, we can get this one. So this one, you must make it becomes Y is equals to MX plus C. So this is a linear, in a linear form. So this is a Lineweaver-Burke equations. Okay, so then another two extra questions are here based on the Lineweaver-Burke equations. What is 1 over X concentration if 1 over V is 0? Then another one is what is 1 over V if 1 over substrate concentration is 0? All right, so we copy the equation okay, from the previous questions. So we like this one. So when the 1 over V is equals to 0, so then you do the simple mathematics. Okay, simple mathematics. So this one you go here, so become negative. No, and this one you go here, become negative. Okay, so then you do that. So that means 1 over substrate concentration is equals to negative 1 over Km. Okay, Michaelis-Menten constant. Okay. Then for the other one, if 1 over substrate concentration is 0, okay, is 0 here. So what happened? That means 1 over V is equals to 1 over V max. So very easy. So then we go to the next question here. Name the type of inhibitor involved in the reactions. So from here, we can see they have the same Vmax. Okay, same Vmax because here is a 1 over V, 1 over Vmax. Okay, then the Km are different, the micromolar constant are different. So this is the characteristic for the competitive inhibitor. Okay, competitive inhibitor. So still remember the previous question here now. X and Y, the Vmax are the same, remain the same, no change. Okay, then the Km change. All right. So therefore, this is competitive inhibitor. There's a type of inhibitor, so your answer must be inhibitor. Determines the Km value for X and Y. So Km value for X, so X is here, X is this one, so -1. So from the previous question here, we know 1 over substrate concentration is equals to -1 over Km. Okay, so -1 over Km is equals to -1. Then look at the unit. This one here is a times 100 times 10 power 3. Okay. So negative, negative, so become positive. Then Km 1 over it. So here 1 over like this one. So here is 1000. So that means it's equals to 0.001 molarity. Okay. Then for Y here is a -2. So same things, -2. Then we make it is a 0.0005 molarity. So remember, Km is a substrate concentration when the Vmax is half. Okay. So therefore, the Km has a positive value with a unit of molarity. Okay, remember positive value. So this is the answer here. Okay, then describe the Km value based on your answer. So just now, this is what we get. The Km value for X is 0.001 molarity, and the Km value for Y is 0.0005 molarity. So here the question is described. So that means in X, okay, in X, the presence of competitive inhibitor doubles the Km value from 0.0005 to molarity to 0.001 molarity. So remember the unit, very important, you must write it. Okay, so that means this is described only. So let's say if I change the question, I modify the question because explain. So the answer will be different. Okay, you need to explain why the Km value for the X is double. Okay, so like here, the Km value is double from 0.0005 molarity to 0.001 molarity in the presence of competitive inhibitor. Not only inhibitor, competitive inhibitor. Then you explain because the inhibitors compete. Okay, so because the competitive inhibitor compete with hydrogen peroxide. Here we need to mention the subtract because the question here say the action of catalase. Okay, catalase. So the substrate will be hydrogen peroxide. Okay, from binding to the active site of catalase, the enzyme name. Then more hydrogen peroxide are required to add in X in order to reach the same Vmax as in Y. Okay, so it's double. Okay, so that's why more. Then next question, what is the relationship between Km value and Vmax value of X and Y? Relationship, X and Y, Km value, okay, and Vmax. Okay, so the competitive inhibition does not change the Vmax because the Vmax still remain the same for X. Okay, so both X and Y have the same Vmax. Okay, so that's why X and Y, both X and Y, they have the same Vmax. But the Kn value increases in the presence of competitive inhibitor as shown in Y. Okay, shown in white here. Okay. Explain the effect of the inhibitor mentioned on catalase. So the effect of inhibitor mentioned on catalase. The competitive inhibitor compete with hydrogen peroxide for binding to the active site of the catalase. The temporary binding, okay, competitive inhibitor is temporary binding only of inhibitor to the active site prevent the hydrogen peroxide bind to the enzyme, okay, bind to the active site of the enzyme. Then the Vmax can be achieved by increasing the hydrogen peroxide concentration. Therefore, the Km value increases. Okay, we go back to this question. Here they say the Km values increases in the presence of competitive inhibitor as shown in white. In the presence of competitive inhibitor is suppose is X, right? X. Okay, so this one is X, shown in X. Okay, extra questions. Let's say if the question they say sketch and label a Michaelis Menten plot based on line reversible plot. Okay, so based on this plot, and then they ask you to sketch this Michaelis Menten plot. So you just get the Vmax, okay, like the Vmax here, okay. So we get it is V1 over Vmax is this one lah. Okay, so then you get the Vmax will be 1.94. Okay, then you have this one is get the you get the 0.97. Okay, then from the what we call this one, okay, then you can get it is 510. So remember the here is a times 10 power negative 4. Okay, here this one I will still follow this one. So remember this is 1 over V, this is V. So that's why here must be negative 9. Here is becomes a positive 1, this is negative 1. Okay, then you draw it. Okay, so you got 1 point here, 1 point here. So you and then another point. So you try to draw it because the question is sketch only. Then after that you label it enzymatic reaction without competitive inhibitor. Then this one is with competitive inhibitor. Okay, so you have the unit, all the points are correct, and label and the graph. Okay, so you will get 2 marks, 3 marks here. Okay, then suggest why the line reversible plot has advantages over the Michaelis Menten plot. Okay, this is also extra questions. Okay, given the line reversible plots give a more accurate. Okay, so the answer is more accurate estimation of Vmax and more accurate information about inhibition. Okay, by increasing the precision through linearizing the data for Km and Vmax. Okay, because here the important one is we linearize the data so that we can get more accurate estimation of Vmax. All right, next question is year 2018. We talk about the enzyme immobilization. So what is meant by enzyme immobilization? So this is definition. So your definition must be a technique to be used to attach, okay? To attach enzyme molecules to an inert insoluble support material or matrix. Okay, remember you must have inert insoluble support material or matrix. Okay. So name the enzyme immobilizing technique shown above. So they're shown in this diagram. So when you see the positive charge, negative charge, so this is adsorption. Okay. So this adsorption is AD, not AB. This absorption is different. Okay. This is penyerapan. Okay. This one adsorption is pelekatan. Okay. State one possible interaction attributed by the technique above. So ionic bonds. Okay. An advantage of an enzyme immobilization is to make the enzymes to be more stable. For example, glucose isomerase is stable at 65 degrees Celsius, for almost a year, but denatures within a few hours at 45 degrees Celsius for free enzyme. Explain how this is possible. Okay, this question is a bit long, but what they want actually is they say why the immobilized glucose isomerase is stable at 65 degrees Celsius. Okay, so for a year, but they just denature within a few hours at 45 degrees Celsius for the free enzyme. So they ask you to explain. So that means this is talk about the advantage of the immobilization enzyme, and then how this enzyme immobilization of enzyme can do this. So through the enzyme immobilization, enzymes would form a multi-point attachment. Okay, so you must have multi-point attachment with their inert support material. So that means the enzyme, they will bind to the inert support or inert insoluble support material or matrix. Okay, so that the three-dimensional shape or the conformation, three-dimensional conformations or the tertiary structure of the enzymes would not change easily. So that's why here they say this restricts the enzymes to change its tertiary structure or the 3D shape or the 3D conformation at 65 degrees Celsius. We get it from the question. Therefore, it provides a stable active site configuration. Okay, a stable active site configuration. Just now we talk about the stable tertiary structure. Now we talk about a stable active site configuration for the binding of substrate at higher temperature. So this higher is 65 degrees, okay, compared to the free enzyme at the 45 degrees Celsius. Okay, so what is the difference between conformation and configuration? So we make it simple. Conformation is the temporary way of the molecule to arrange itself. So that means the shape or the structure of something. Then the configuration is an arrangement of parts or elements in a particular form or figure. So they just parts only. Okay. These definitions come from the Oxford Dictionary lah. Sure, in chemistry, actually configuration they can have many different meaning. Okay, in different topic. Okay, so if you want what we call the more or better, so you just write active site shape or the shape of active site. So everything you use shape or the structure. Okay, so what is multi-point attachment? So look at here lah. Free enzyme. Okay, so this is immobilized enzyme. So they attach to the inert insoluble support material. So they have multi-point attachment here. So this multi-point attachment, so when the temperatures increases, so like just now in the 65 degrees Celsius, so for the free enzyme, the enzyme conformations is changed and the active site configuration is changed also. So, but for the immobilized enzyme, they have a less distorted they may pull or twist, okay, because the enzyme structure, okay, or we can say the enzyme conformation or the tertiary structure or the three-dimensional shape of the enzymes are stabilized, okay, stabilized by the what we call the multi-point attachment to the inert insoluble support matrix. Okay, how enzyme immobilization could make enzyme more stable at high temperature? So this question is a simple question. So like just now, they have the same meaning. So through the enzyme immobilization, enzyme would form multi-point attachment with its support matrices. Okay, can write inert support matrices, inert insoluble support matrices also can. Restricted the change of enzyme shape. Okay, that means this enzyme mobilization restricted the change of the enzyme shape of three-dimensional conformation at high temperature. Therefore, each provide a stable structure of active site. Like just now, if you don't want to say configuration, you can write a stable structure of active site or the stable shape of active sites. Okay, not the stable enzyme. Okay. then the immobilized enzyme will not be denatured easily. You cannot say not be denatured only. You cannot say will not be denatured at high temperature. No, will not be denatured easily at high temperature compared to free enzymes. Okay. All right. Then when we talk about the enzyme immobilization, so we will need to talk about the biosensor. So the biosensor shown below make use of the enzyme urease to measure urea in either blood or urine. So this is the equations given. Okay, so this is the diagram of the biosensor. So you see there is the solute, okay, including the urea, and then they have partially permeable membrane here, right? Then they have immobilized urease. Okay, this is the product. Okay, the product ammonium ions, and the transducer, amplifier, and display. So the amplifier actually they plus the processor microprocessors inside here All right, definition. Okay, like here they got one question said define what is immobilized ureas. So this is the general definition for immobilized enzyme. Enzyme that are trapped, attached, confined, or restricted to a fixed solid, maybe insoluble structure or support matrix. Okay, or inert. So for the support matrix, there can be organic support matrix, for example, cellulose, agarose, alginate, resin, polyacrylamide, or inorganic matrices. Inorganic matrices, they are silica gel or alumina. So just remember the simple one. For example, here support matrix for organic, alginate, okay, alginate or cellulose. Then inorganic silica gel, okay. So here define what is immobilized urea. So this is specific one because they give you the ureas. So urease that are trapped or attached, confined, or restricted. That means you either one only to a fixed solid insoluble structure, okay, or the inert support matrix in a biosensor. Okay, because later we have to say it in a biosensor lah. So suggest a substance that could be used to immobilize the urease. Okay, so this one refers to this support matrix. So like just the question says is a substance, that means one only lah. So since the question didn't mention is organic or inorganic, so you can choose either one. So remember the question said one, so that means one. Example is given. Explain the significance of the partially permeable membrane. Why the partially permeable membrane is so important? Okay, allows the urea and smaller solute to pass through. That means they only allow the urea and smaller solute to pass through the partially permeable membrane in order to increase the probability of urea binds to the immobilized urease. Okay, so the important here is to increase the improbability. Okay, allow this one and increase the probability. Right. So we can use partially or selectively. Since the question already give the partially, so we just follow it. Cannot use semi-permeable. Okay, explain why other solutes do not affect the result. So this one is talk about the specificity of enzyme. Not the specific enzyme, and specificity of enzyme. So only the urea can bind complementary, okay, complementary to the active site of urease to form urease urea complex, the enzyme substrate complex, and then form ammonium ions. Okay, and urea or other solute unable to bind complement to the active site. Okay, we go back to here. Carbon dioxide. All right, yeah, this one for you. And carbon dioxide. This one not urea and carbon dioxide. Okay, okay. Say other solute unable to bind complementary to the active site of urease. Okay, so this one we talk about the substrate specificity of enzyme. Okay, so this is referred to specific substrate, not specific substrate, and then not the specific enzyme. Okay, describe the function of the transducer. The function of transducer is to detect the presence of ammonium ions and convert the chemical signal. This ammonium ions is a chemical signal into electrical signal. So we cannot accept the chemical energy or electrical energy here. It is signal, a signal, okay? States two advantages of using a biosensor rather than a chemical method, okay, to measure the concentration of urea. Small quantity of sample is required and result is obtained rapidly. So only two. Huh? Or you can write ease of use, small and portable, reusable for continuous monitoring. Okay. All right, next question. The diagram below shows a biosensor used to detect blood glucose. So similar, here can see there is a partially permeable membrane. Then they have immobilized enzyme, okay, immobilized glucose oxidase. Then they have the platinum electrode here, okay, extra one, platinum electrode. And then the transducer, amplifier, processor, okay, digital readout. Okay, then the reactions equations given, okay, oxygen, glucose plus oxygen, that means glucose plus oxygen. And then you have the glucose oxidase, okay, and immobilized. Then convert into the gluconic acid and hydrogen peroxide here. Okay, then the platinum electrode will convert the hydrogen peroxide, okay, or oxidize it or split it into hydrogens, ions, oxygen, and electrons. So does that mean these three products will be here? Okay. Now, once you already understand the question, now you can answer it. State the characteristic of the membrane. Since here they only use membrane, so the characteristic is partially permeable membrane or selectively permeable membrane. We cannot accept semi-permeable. Okay, explain the significance of the membrane. So similar answer are given. Okay, allows glucose and smaller solute to pass through the partially permeable membrane in order to increase the probability of glucose binds to the immobilized glucose oxidase. Okay, we have immobilized. Decomposition of hydrogen peroxide by platinum electrons liberates hydrogens, ions, oxygen, and electrons. So there are three products here. State which product is detected by the transducer. Okay, so they'll be detected by the transducer is electrons. Okay, why? The result will be less accurate because hydrogen, ion, and oxygens are also found in blood. Okay, if we detect the presence of hydrogens, ions, or oxygen. However, only the electrons released, okay, are the product from decomposition of hydrogen peroxide that can be used to be detected by the transducer. Okay, can be represented or indicated the amount of glucose. Okay. Describe what happened to the product detected by transducer until displayed as a digital readout. So the main this question that describes start from here. What happens here in the transducer to the end? So the transducer detect and measure the presence of electrons and convert it into an electrical signal. Okay, the electrons here is the chemical signal. Okay, they convert into an electrical signal. Then this electrical signal actually is very small. Then it is amplified. Okay, amplified by an amplifier. Okay, amplifier here before it is fed into the processor, the microprocessor. So this microprocessor, they will process the signal, interpreted, and display it in a suitable readout. Okay. So this is here they talk about we discuss about the function of the transducer and the function of the amplifier, okay, the microprocessor and the readout, okay, all together. Okay. Then in year 2015, there is one question: describe the mechanism of an enzyme action based on induced fit hypothesis. So this is induced fit hypothesis. According to the induced fit hypothesis, the active site of an enzyme is not a rigid receptacle for the specific substrate. Okay, so you see the active site of an enzyme is not a rigid one. Okay, it's not a rigid receptacle for the specific substrate. So the active site is being flexible to the substrate, not the substrate is flexible to the enzyme. Okay, then as the substrate binds to the active site of the enzyme to form enzyme substrate complex, so what happens to the enzyme? The binding causes the enzymes to change its shape slightly. See here. Okay, see here. And they already changed. Okay, slightly lah. So that the active site enfolds or distort the substrate and hold it in place. Then the shape of the substrate, you see the shape of the substrate also change slightly. So that this can enhance the ability of the enzyme to catalyze, okay, the chemical reactions. So the chemical reactions are, they will break the existing chemical bonds in the substrate and form the new chemical bonds. So this one actually is the interactions or the catalysis. Okay, so this one actually how they break is occur between the chemical groups of the substrate and R groups of the amino acid in the active site of the enzyme. So we know the active site actually they consist of just several amino acid, okay, where the R groups are normally they are polar or charged. Okay, then product is formed and released. So once they form the product, okay, so the products will be released, okay, from the active site of the enzyme. Then the enzyme reverts to its original conformation. Okay, this is the original conformation. Then the enzyme is available for the next reaction. So this is induced fit hypothesis, how to describe the mechanisms. Okay, remember. Then the next question is explain how an enzyme accelerate a chemical reactions, year 2015. So here is how enzyme accelerate. Okay, so here I use the induced fit hypothesis. An enzyme accelerate a chemical reactions, so by lowering the activation energy. Okay, so when you can write this by lowering the activation energy, you get 1 marks here lah. Then a specific substrate bind complementary to the active site of the enzyme. So we must always mention active site of the enzyme to form enzyme substrate complex. This provide a conducive microenvironment in the active site for the reactions to occur. Okay, so the microenvironment here. So like just like in the previous question, if you want to apply this sentence also can, no problems. Okay, microenvironments. So actually what happens to the microenvironments? Okay, same like just now we have discussed, the interactions or the catalysis occur. All right. So a brief covalent bonding is formed between the active site and the specific substrate. Okay, like just now I said, the chemical group of the substrate interact with the R group of the amino acid in the active site of enzyme. Okay, between distorting the substrate and thus reducing the amount of free energy required, okay, to be absorbed to form the enzyme substrate complex. So this is to lowering the activation's energy. Okay, then these catalytic reactions convert the specific substrate into product. Like just now in the previous question, let's say you want to apply this sentence, also can to break down the existing. Let me here to break down the existing chemical bonds, okay, in the substrate and form the new bonds in the product. Also can. Okay. So the product is released from the active site and the enzyme is available for the next catalytic reactions. So see here, I provide the alternative way, okay, how to answer the similar question. Okay, but this one is explain how an enzyme accelerate. Okay, they accelerate. But the previous one, they say induced fit. So that means here we must apply the induced fit knowledge. Okay, so the induced fit information is talked about where the active site of the enzyme change slightly and the shape of the substrate also change slightly. Okay, and then here they focuses on the how the enzyme they can accelerate the chemical reaction. So we say by lowering the activation energy, and actually how, okay, how the enzyme can lower the activation of energy. So here they need to focus to the conducive microenvironment. So actually what will happen to the to this conducive microenvironment? So it's like this one lah. Okay, so actually they are similar, okay, to the previous question, but this one they are not focused on the induced fit hypothesis. If you apply the induced fit hypothesis in these questions, also no problems, okay, because their focusing is on this. All right. Year 2015, describe the significance of Km, microris maintain constant value in enzyme catalyzed reactions. So they say describe the significance. The significance are not the relationship between this one. So microris maintain constant is a measure of the substrate concentration required for effective catalysis to occur. So required for effective catalysis to occur. The Km is determined as the substrate concentration at half Vmax. Enzyme with low Km has a high affinity toward its specific substrate. Therefore, the enzyme will bind to the substrate more tightly to form enzyme substrate complex. So the enzyme functions effectively and requires low substrate concentration to achieve maximum reaction velocity. So this one, if we relate this one to the conducive microenvironments, so they provide a more conducive microenvironment. So that at low substrate concentration, the rate of reaction can achieve the maximum or reach maximum rate. Whereas the enzyme with high Km has a low affinity towards its specific substrate. So the enzyme binds to the substrate, but is more weakly or less readily. So to form enzyme substrate complex. So because they bind to the substrate more weakly, like just now we see in the free energy diagram in the transition state. So most probably the substrate cannot form product. They will return to the substrate. Okay. So higher substrate concentration is required to achieve maximum reaction velocity. If you remember or you understand about how we calculate, we get the microris maintain constant, they got K1, K2, this one, where they form the enzyme substrate complex. So from the substrate and enzyme to enzyme substrate complex, the reaction or the process is reversible. Okay, reversible. Okay, so that means from in the transition state, the enzyme substrate complex, the substrate still maybe they either form product or they can return to the substrate and enzyme. Okay, so no product is formed. So it means that enzymes with low Km are able to function at maximum velocity at low concentration of substrate, whereas enzymes with high Km only function effectively at much higher substrate concentration. So from there, if we get a list where the enzyme and the respective Km value, so we from the Km value we know the enzyme with very low Km, so they can achieve maximum rate of velocity faster, at low concentration of substrate lah. Okay, compared to the higher Km. Okay, next question. Describe the effect of low temperature such as 5 degrees Celsius on enzyme action. So this one talk about the factors. So at 5 degrees Celsius, kinetic energy processed by substrate and enzyme is low. Here we talk about kinetic energy. So when it's low, the enzyme is said to be inactive, not denatured, it's inactive. Less effective collision. So we talk about effective collision. Okay, less effective collision between substrate and enzymes to be occur, then less substrate binds complementary to the active site of the enzyme to form the enzyme substrate complex. That means a less enzyme substrate complex formed and less product is formed. Then at the end, the rate of enzymatic reaction is low. So this is at low temperature. How about the temperatures at 65 degrees Celsius? So we know at 65 degrees Celsius, the enzyme is, most of the enzymes are denatured. So at 65 degrees Celsius, kinetic energy possessed by the substrate and enzyme is high. Okay, then the bonding in the enzyme, such as hydrogen bonds, disulfide bonds, are less stable and broken down, okay, due to the high kinetic energy. Then the tertiary structure of enzyme is changed. The configuration of the active sites also change. Then the enzyme is said to be denatured. The specific substrate unable to bind to the active site of the enzyme because the configuration of the active site is changed. Then less or no, here we can say less because it's already at 65 degrees Celsius. So enzyme substrate complex does not form. Then no product is formed. The rate of enzymatic reaction is stopped. So it's zero lah. Okay. No, the rate of enzymatic is zero. The enzymatic reaction is stopped. Okay. Okay, zero. Unless the question change from let's say from 40 degrees Celsius to 65 degrees Celsius, so we can say less substrate complex is formed. Okay, then less product is formed. But when they reach the 65 degrees Celsius, so everything is stopped. The next question, describe the three types of core factor and give one example of each factor, core factor. So there are three: coenzyme, prosthetic group, and activator, or we call metal ions. Okay, for the coenzyme, so you need to describe what is coenzyme. It's an organic non-protein molecules. Okay, organic non-protein. They bind to the active site of enzyme loosely and temporarily. This is coenzyme. So examples is oxidized NAD, oxidized NADP. Since they say give one example, so you must only write one example lah. Prosthetic group, also organic non-protein molecules, but they attach permanently to the active sites of the enzyme. So for example, heme in catalase and peroxidase, or can say FAD in succinate dehydrogenase. Here the heme is in the enzyme. I cannot say heme in hemoglobin. Okay, because the question is refers to the enzyme. All right. Then activators or metal ions, they are inorganic molecules or inorganic particles. They bind temporarily to the active site of the enzyme. So the example is this one. Magnesium, you can write the magnesium, okay, or magnesium's ions is in kinase, okay, kinase. Then in year 2020, also describe three types of core factor and their role in enzymatic reactions. So similar. So here, okay, they say describe three types. So it's mentioned this one. Okay, and then participate in reaction by donating or accepting certain chemical groups, electrons or atoms between enzyme substrate. Okay. Then the core enzymes, you explain what is that like just now. Okay. Then because here they say their role in enzymatic reaction, so it's describe their role. Not give one example only. So you need to say, for example, oxidized NAD in dehydrogenase. Okay, play the role in dehydrogenase, receive electron and hydrogen atoms to form reduced NAD. So reduced NAD, you better write the full form. Okay, not the simplified form. This is if just NADH is simplified form. This one is full. The accurate one, the correct one. Okay, so coenzyme, this one apple enzyme, coenzyme to have hollow enzyme, the functional enzyme. This one I think you should know. Okay, prosthetic groups attached tightly and permanently, so the same. So and this one we just look at here, iron in heme group. Okay, of catalase receive oxygen. So we talk about the heme group lah, receive oxygen atoms from hydrogen peroxide to form oxygen molecule. Okay, so not in hemoglobin again. We talk about enzyme. Then the activator metal ions, okay, same. So this one magnesium's ion in for kinase and then calcium's ion for thrombokinase. Just one only enough lah. One example for each cofactor. Okay, explain in year 2019, explain the mode of action of a competitive inhibitor. So since a competitive inhibitor, so it's structurally similar to the specific substrate. Then the competitive inhibitor competes with the specific substrate for binding to the active site of the enzyme. So important, everything that I highlight here. Okay, and then the competitive inhibitor, since it says bind temporarily, here got bind temporarily or not, so bind non-covalently to the active site of the enzyme. So it's temporary. Now I can add binds temporarily and non-covalently to the active site of the enzyme. Then this prevents the specific substrate to bind to the active site of the enzyme. Okay, you must have this because the binding of the competitive inhibitor to the active sites prevent the specific substrate to bind to the active site. So no enzyme substrate complex is formed and no product is formed. And then the enzyme also unable to convert the competitive inhibitor into product. Okay, then the competitive inhibitor does not permanently inhibit the enzyme. As I told you just now, they are non-covalently bind, or they are permanent, they are temporary bind. So this competitive inhibition is temporary and known as reversible inhibition. Okay, so every point that I show here is important. Okay, see, simple, compact. So the effect of competitive inhibitions can be overcome by increasing the substrate concentration. These are extra lah. When the amount of substrate is relatively greater than the competitive inhibitors, that means more substrate occupied the active site of the enzyme most of the time. Probability of substrate binding to the active site of enzyme to form enzyme substrate complex increases. Then the rate of reactions increases as the increase in the substrate concentration. You see, when you increase the substrate concentration, so that means probability for the substrate or for the specific substrate bind to the active site increases compared to the competitive inhibitor. So that means more enzyme substrate complex form, more products will be formed. In year 2020, now they use this question. Aminoglucosidase is an enzyme that breaks down starch to glucose. Explain why hydrolysis of starch stopped when heavy metal ions are added to the enzymatic reaction. You see, some question, they like to give you a situation. So if in this question, when you want to answer this question, you must use the information given in the question. Okay, like the enzyme is aminoglucosidase. The substrate is starch. The product is glucose. Here, heavy metal ions is what we call irreversible inhibitor. Actually, irreversible inhibitor is not in your syllabus. Your syllabus is reversible inhibition. Okay, you can refer to the syllabus. Heavy metals, but anyway, we can answer it. Heavy metals ions such as mercury ions, okay, give the example lah. Cadmium ions and lead ions, they still got many lah. So are irreversible inhibitors. So actually, this one, these metal ions, heavy metals ions, they have no similar structure to starch. Okay, they have no similar structure to the substrate. Okay, so here you must write heavy metals ions have no similar structure to starch because the question is given here. Then the heavy metal ions do not compete. When you say no similar structure, so that means do not compete with starch for bindings to the active sites of aminoglucosidase. Please use full sentence and not only do not compete. What do not compete? Okay, so heavy metal ions do not compete with starch for bindings to the active site of aminoglucosidase. Okay. Then heavy metal ions strongly bound to the sulfhydryl groups on cysteine amino acid of aminoglucosidase. Okay, so here this is the R group. Here they got SH. Okay, sulfur group. So when's the mercury ions? Okay, mercury ions they come. So they will form covalent bonds, not disulfide bonds. Disulfide bonds is formed between the sulfur group and the sulfur group. But this one, you see, they form covalent bonds, okay, with the sulfur groups, okay, in the amino glucosidase. So when they form these covalent bonds, they will alter, okay, or change the tertiary structure of aminoglucosidates. Okay, you see here the structure, the tertiary structure or the 3D conformations of the aminoglucosidase change, and also change its active site configuration. See the active site configuration here also change. All right. So extra information for you. Mercury ions also binds to the phosphoryl, carboxyl amide, and amine groups. All right. So they seldom bind to the active site. Okay. In research, the research show that most of the heavy metal ions, yeah, especially these mercury ions, and also others heavy metal ions, they mostly binds to the sulfur groups. Okay, and this one. Okay, then the starch unable to bind to the active site to form aminoglucosidase starch complex. No glucose is formed. So for irreversible inhibitor, actually they binds to the enzyme, then they form covalent bonds. So it is permanently or tightly, okay, tightly. All right. So the hydrolysis of starch stops as all the aminoglucosidases are inhibited by the heavy metal ions. Covalent bond is very strong. It's a strong bond. Strong bond. So it's tightly. The irreversible inhibitor bind tightly to the enzyme or the aminoglucosidase in this question. Okay. In year 2017, name 5 classes of enzyme and state its function according to the International Union of Biochemistry, IUB. Now the IUB becomes IUBNB lah. International Union of Biochemistry and Molecular Biology. Okay. Classes of enzyme are hydrolases, isomerases, ligases, oxidoreductases, and transferases. So, but the question is 5 classes, so you just give them 5 classes enough lah. So here I show you 6 classes lah. Okay, so you write hydrolases, catalyze. So remember you must have catalyze. No, cannot use convert. Okay, and so on. So hydrolases catalyze hydrolysis of molecule by addition of water. Isomerases catalyze rearrangement of atoms in a molecule from one isomeric form to another. Liases catalyze non-hydrolytic. Okay, this one and this one is split, but this one hydrolysis use water. These liases are non-hydrolytic addition or removal of part of substrate molecule, thereby creating double bonds. They remove part of the substrate, but no addition of water. Ligases catalyze the joining together of two molecules coupled with the hydrolysis of ATP. Okay, ligases, hydrolysis of ATP, remember. Oxidoreductases catalyze biological oxidation reduction reaction. So redox by the transfer of hydrogens, oxygen, or electrons from one molecule to another. Transferases catalyze the transfer of a functional group from one substrate to another. Okay, so just write 5 classes. So this question is more on the, it's a factual question. What is meant by enzyme immobilization? Okay, so it's a technique to be used to attach or fix enzyme molecules to an inert insoluble support material. So this is talk about enzyme immobilization. It is a technique. Just now they got the definition is immobilized enzyme. That was referred to the enzyme. Okay, explain what is biosensor. two applications of biosensor and one example of its usage. So what is biosensor? Biosensor is an electronic analytical device which uses immobilized biological materials. So these immobilized biological materials such as enzymes, they mean the immobilized enzyme or immobilized antibodies. So since you only learn immobilized enzyme, so we can remove these antibodies. Okay, so that means the biosensor is an electronic analytical device which uses immobilized biological materials such as enzyme, okay, to detect, okay, or which uses immobilized enzymes to detect chemical compounds and convert it into an electrical signal. See, from the chemical signal to electrical signal. Okay. Application. No, they explain what is. So the immobilized enzyme are specific in detecting the particular molecules in a complex mixture. Then the reaction between immobilized enzyme and the substrate that produce a change. So the change can be release of heat, change in pH, flow of electrons, production of new chemicals, and so on, which is converted into an electrical signal by transducer. Okay, so this is the function of transducer. Okay, they can detect either all of these, okay, the change. Then the electrical signal is amplified, interpreted, and quantified to display a digital readout. So this is explain what is biosensor, okay, and then how's the biosensor function. Okay, then the application. Biosensors can be applied in medical field, food industry, environment, military, and public security. So just mention two applications enough because the question here is two. Then one examples of each usage. So this is a 7 marks question. So 1 plus 2 is 3. So that means 4 is for the explanation on the biosensor. So the example is only one. Okay, if possible, we related lah. Like let's say here you mentioned the medical field. So here the example is the medical field. Okay, if you talk about environment, so here we talk about the environment. Environment. Oh, here didn't mention the environment. Environment can be the, for example, the presence of pollutant. Okay, the pollutant in river. Okay, or the heavy metals. All right, here, for example, blood glucose bar sensor. So in medical field, okay, medical field. So the example is the blood glucose biosensor or the biosensor can be used to detect blood glucose level of a diabetic patient. Okay, so here I give you several examples lah. So you can look at here, urea, this one just now we also did a question before. Hemoglobin biosensor used to detect the hemoglobin level in the blood sample. And then the biosensor also can be used to detect the pathogenic bacteria in food. And then the biosensor used for detecting the nerve agent in public. So this one for the public security, okay, against the terrorism. And then if in future, maybe you can create a biosensor can detect the presence of coronavirus, maybe. Okay. So resin beads are used as metrics in entrapment techniques for enzyme immobilization technology. States the role of resin beads and advantages of enzyme immobilization. Year 2016. So here they give you the enzyme immobilization technique is, which is entrapment. Okay. State the role, okay, and advantage. So resin is an inert, okay, insoluble support matrix, and then it's used to trap enzyme within the resin. So the enzyme are trapped in the resin, not to attach. Okay, you must remember entrapment is to trap, not to attach. So in entrapment, the enzymes are not physically or chemically bound to the inert matrix. Then the advantages, so that means at least two lah, at least two advantages given. So enhance, that means write advantages of, say, enzyme immobilization. Okay, please use complete sentence. Enzyme immobilization enhance the stability and activity of the immobilized enzyme. Okay, or the immobilized enzymes can be reused, so can be used in continuous fermentation. Cost of commercially use of enzyme can be reduced in mass production. Okay, if you want to talk about the cost of immobilization enzyme or biosensor, so it's lower or cheaper, but you must have in mass production. Okay, only in mass production or bulk production you can get the cheaper cost. And easy separation or the purification of immobilized enzyme from the product. Okay, can be done easily. And then the downstream processing cost, okay, the cost for the downstream processing, that means including the purification here, can be minimized. And that's all for this chapter 4. So I think there are a lot of information for you. So good luck, okay, and thank you for listening. Bye.